Our next speaker, Dr. Judith Steen, is a prominent figure in the academic and medical community, currently serving as a distinguished faculty member at Harvard Medical School. Her work in proteomics and neurodegenerative diseases has not only earned her widespread recognition, but has also influenced the outlook on these critical areas. Dr. Steen holds a PhD in biochemistry, biophysics, and molecular biology, and was a postdoctoral fellow in proteomics and cell cycle at Harvard University. She has consistently demonstrated a commitment to advancing our understanding of cellular and molecular processes. Her research is dedicated to forging new qualitative and quantitative methodologies that intersect proteomics and transcriptomics. Dr. Steen research aims to utilize molecular information derived from our quantitative proteomics measurements to mitigate neurodegeneration. We are honored to welcome Dr. Steen to share her expertise with us. Please welcome Dr. Judith Steen.
Really wonderful time for me here, meeting all of the students. I actually had a bit of a moment with some of the best students at this school in the CS class. So I was very excited to interact with this incredible human capital here. And I'd like to thank Dr. Sristava for organizing this and for all of the incredible students that have been working really hard to make this happen. So this was something that one of my students, my PhD students, drew in the lab just to get an overview of what we were doing in the lab. So what we're trying to do in our lab is to understand why neurons die. And this could be due to aging, injury, genetics. But we also want to understand how we can keep them surviving and how they actually differentiate and maintain themselves. And because it's part and parcel of understanding how the brain works. So there's a major unmet need in neurodegenerative diseases, partly because we are aging rapidly. The populations today, and especially the developing countries, are aging rapidly. And one of the biggest aspects of this is the fact that, by the time you reach 85, something like 30% of us will have some form of dementia. So how do we age in health without forgetting who we are? I actually had a grandmother who lived with us, and I watched her as I grew up forget who she was, and I think it was the most painful thing to watch for my mother, and for her, because she couldn't remember so much as she grew older, and the question is, how much is life worth living after forgetting who you are? So right now, early diagnosis is not possible, and I'm talking pre-disease diagnosis. And there is no cure for Alzheimer's disease or any other dementias. All we do is mitigate the actual symptoms. So one thing that we know is that 75% of all neurodegenerative diseases or dementias actually involve tau. However, we do know that there are certain genetic predispositions that result in Alzheimer's disease, but they're actually risk factors. Only 2% or less than 2% are causal. So if you think about the ApoE4 gene, if you have 60% of all Alzheimer patients have at least one allele of ApoE4. However, just because, and this goes back to what Dr. Kannan was talking about yesterday, there are 4 aspects in your life that you can control. And if you don't, diet, if you have some trauma, if you have infections, toxins, lifestyle, including tobacco, it actually affects whether or not you develop dementia. And it's been studied in the case of ApoE4. So the question is, how does this impinge on your proteome? Because the results of these particular stressors in your life are found on your proteome. They're not necessarily found on your genome. So using proteomics, we can identify proteins, we can look at the structure, we can quantify them, we can look at interactions, we can look at post-translation modifications and study the kinetics and dynamics of modifications as well as proteoforms. And I'm very glad that we had this proteoform discussion before, because I don't have to talk about it as much. So in the last 5 to 7 years, it's been quite well established that tau is a prion-like molecule. And there have been several studies recently, including a very shocking paper last week in Nature, where growth hormone that has been derived from cadavers has resulted in early onset dementias in the subjects that received the growth hormones. So, and this is purified growth hormone. It's really hard to purify proteins to 100% purity. These things will follow you everywhere. So I think we have to be really careful about that. So what we know basically is that although tau has very important functions in stabilizing microtubules, and I had a really interesting discussion with my former mentor who actually discovered the molecule, Mark Kirschner, back in the 80s. And he said that it was a really interesting story, but at that point in time, nobody knew about splicing, so they had these 6 bands on the gel, and they couldn't explain what it was, but they knew it was the same protein. Anyway, so what we know we can possibly do to stop it, to stop this prion-like form, is to reduce expression. We can regulate PTMs on it. We can decrease aggregation by interfering with the aggregation, make molecules that can prevent the tau forms from aggregating. We can accelerate degradation of the aggregates. We can mitigate spreading, but we first have to understand what causes the aggregates to form. What initiates the tau prion-like fold? And the other aspect is, how does tau spread from neuron to neuron? Now, there have been several drugs that have been tried in this arena. However, the antibody drugs, for example, have been made only to primary sequences, and there have been a couple that have been made to post-translation modifications, a couple of phosphorylations. When we started this study, there were only about 30 post-translation modifications mapped. And most of them were studied using antibodies and not in depth with mass spectrometry. So the question we were asking is, there are so many uncertainties with respect to tau. What is the event discovery? What is the cause and effect? How can we build better models and evaluation systems and develop diagnosis and therapies? That was the goal. So our strategy to do this was to investigate the whole problem with mass spec-based proteomics. So when you look at the current Alzheimer's disease development pathway, what happens is you basically start with a model where you've knocked in specific genes that have been associated with the disease. You then study this model, you do preclinical, you identify a pathway and a target, you do your preclinical testing in this model, and then you move into the human. However, the issue is, are these models actually reflective of the human? So the question we were asking, are the models reflective? Do we have enough biomarkers that can stratify the subjects pre-treatment? And do these biomarkers really measure target engagement and disease progression? Right now, they're using cognitive tests to determine whether or not somebody's progressing. However, people who enter these trials are pretty smart, even if they have early signs of dementia, and they do learn. And so if you're giving them the same test day after day, it doesn't help in determining your negative and positive controls, right? So one of the first things we did was to evaluate the most common common mouse models used for preclinical testing. And lo and behold, they're not really reflective of human disease. They don't even reach the symptomatic stages. In terms of the post-translation modifications, they don't show the same symptoms as the patients as well. And the sites of modification are different because most of these models have mutations. And the mutations affect the fold and also the actual profiles of the tau forms. And this work was done by an incredible PhD student in my lab. She's got 2 other papers and just published in Nature Protocols on her work for very low amounts of sample in flexi-tau. And Patrick Van Zam and Arthur Viode. So basically this particular study showed us a lot of different things. And so we decided to use large human data sets to determine what good targets were and to understand the disease. And use the human fluids and brain tissues in order to find targets and biomarkers. And then we are also developing new mouse models to do this and cellular models for drug screening. And we want to find biomarkers that actually measure the target engagement and disease progression and stratify these subjects first using the biomarkers. So one of the first, the next studies we did was to look at tau PTM profiles to identify heterogeneity and stages of Alzheimer's disease. This work was, as I said, initiated by Valtraud Meyer, who developed the flexi-tau assay, and she was helped by Henrik, Mukesh Kumar, Xiao Jun, Chris, and Hannah was also involved. So, what we did was we studied a cohort of human subjects who ranged from BRAC0, which is no tau aggregates, to BRAC6, which is the most severe form of Alzheimer's. And we asked the question, what are the molecular features of pathologic tau? And can these features differentiate between the symptomatic and asymptomatic patients? And how do these PTMs behave over the disease progression? So, the snapshot of our workflow is that we take these Alzheimer's disease patient brain tissues. We look at more than one area of the brain. We look at insoluble, soluble. We developed a seeding assay. This is a FRET assay to understand whether the aggregates are seeding positive. Do they have prion-like behavior or not? And we basically look at the tau in the aggregates. And here I'll show you that the tau is the most abundant for a protein in these particular aggregates, but I want to point out that there are several other proteins that are also found in these aggregates. So I strongly believe by now that they influence each other, and we have a paper in PrEP that shows that there are interactions between synuclein and tau, for example. And then we use targeted and non-targeted assays to study both tau and the global proteome to understand the actual mechanism and the pathways that are involved in putting or writing these post-translation modifications on the tau. And in the controls, we also look at the eraser molecules that might be removing the tau to see if we can enhance those eraser molecules as well. So so flexi-tau basically is a method by which it's very unbiased and it's very simple. So basically what we do is we add a heavy isotope label standard with a flex peptide that allows us to quantify tau absolutely. And basically, we spiked this heavy labeled standard into the PrEP, the sarcosyl PrEP, and it follows through the whole digestion protocol. And basically, what we do is we monitor, it's completely unmodified. We monitor the unmodified peptides, not the modified, but the unmodified. And we quantify those over the MS analysis using Skyline. And thank you, Dr. McLean, for doing the Skyline workshop yesterday. So basically, what we do is we look for peptides where we see a decrease in the modification extent. And then we find the modified species, and we can then do targeted assays for the modified species as well. So why is the absolute amount of tau really important? It's really important for drugging your tau, for finding biomarkers. If you're making a PET reagent, if you don't know how much or what the concentration of the pathologic tau is in your brain, you can't develop a drug or a molecule that targets that effectively, and you don't know which binding affinities you need for those particular PET reagents unless you know the absolute quantification. So the other aspect is there are 6 isoforms, as I mentioned, and Mark Krishna at the time in the 80s, because splicing hadn't been found, was it might be 70s, late 70s actually. So we quantified all of the different isoforms, and we found that in the insoluble tau, the 0 isoform is the most abundant compared to all of the other ones. So the 2N is incredibly low. So 1N and 0N are the most abundant. And there is a 3 to 4 hour distribution that's almost equal. So in total, we mapped 95 modifications. Basically we looked at the patient frequency, because this is really, really important, because mass spec is incredibly sensitive these days, and understanding what the patient frequency is across the patient population also helps you to hone in on the most important modifications in your pathologic fractions. So there were the usual suspects for which there were antibody detection methods, but then there were several other ones. So which ones are important? So by doing hierarchical clustering, we basically were able to find 4 different groupings. The first was a control group, where we did see some physiological phosphorylations. So although in the field it's been suggested that the modifications cause the removal of the tau from the aggregates, this is not actually true. There are some physiological aggregates, physiological modifications on the non-aggregating tau. And then the second control group had, and then there were 2 AD groups, one with more modifications than the other. So when we looked at the staging of those particular groups, we found that these had the lowest Braak stages, these controls. These had slightly higher Braak stages and also contained some symptomatic subjects here. Whereas the highest Braak stage was found in with the highest numbers of modifications. So basically, phosphorylation occurs early in disease, but it's not indicative of symptomatic disease. And you can basically erase some of those if you can enhance some of the particular phosphatases involved. Tau acetylation and ubiquitination are associated with symptomatic but not asymptomatic disease. And you can see here that in the microtubule binding domain, you get among the symptomatic patients. So every single row here is a human subject, by the way, and every single column is a PTM. So here, for example, you can see that the black are the AD patients that were clinically and pathologically diagnosed. They had these modifications in these microtubule binding domains that were either acetylation or ubiquitination. And what we've also noticed from this particular study is the fact that you get a very ordered progression of post-translational modifications. It doesn't happen haphazardly. So there seems to be, if you look at the pathways at the different stages, there seems to be very specific means of aggregating. So when we look at the flexi-tau, what was really evident was the fact that the C-terminus is completely modified and cleaved. The C-terminus is, whereas in the controls, the C-terminus is completely unmodified, as is the N-terminus. And in the most, the highest Braak stages, you see that the C-terminus and the N-terminus are completely modified. And what does this tell us? It gives us a list of PTMs and regions of the tau that are specific, that are good to target for antibody development. One thing is if you want to develop an N-terminal antibody, which was like goracenimab, which failed drug trials, you want to avoid the N-terminus. So in summary, we understood how the disease progresses. And basically, we know that there is a very specific order in which these events occur. And we've also identified very specific modifications that are associated with the prion-like form of tau, which we are targeting for therapeutics. The other aspect is we know through our cryo-EM and PTM studies of some of these tauopathies that there are specific modifications that affect the structure. However, most of the cryo-EM people are not completely convinced, and because the molecule is heterogeneous, and you don't see the densities at every cross-section of these molecules. So because of that, some of these, they see the densities in some of the aggregates, but not in some of their structures, but not in all of them. So another aspect that we've been looking at is the pathways, and we've modeled this in cell cultures. We have a high throughput system for basically screening drugs, and we use mass spec for this. Whoops. We use mass spec for this. So what I can say is we are approaching these challenges head on, and with a wonderful team in the lab, we are also looking at this FlexiQuant framework for other proteopathies. And I'd like to thank all of the funding agencies that have given us money, especially the Tau Consortium, who set me off on the